So we have here a laboratory test for you to diagnose a patient with a cytomegalovirus. So remember that when you are collecting your blood, it should be performed in a septic technique. Your sample could eventually be a serum sample. Could also have here your anticoagulated blood or your plasma. Collect that one in either your EDTA or you could also have your CPDA1 as your anticoagulant. Okay, the next one, we have here your Epstein-Barr virus. Okay, the Epstein-Barr virus also belongs to herpes viridae. So this is also a double-stranded DNA is an envelope na virus. This one could be transmitted here by contact with your nasopharyngeal secretions. That's why it's being called here as your kissing disease. Could be transmitted also here by blood transfusion. Could also have here the transplacental. Okay, so the virus here basically try to suppress the host immune response by the following mechanisms. Number one, we have here signal transduction, also try to inhibit the cell cycle control, regulation of the gene expression, post-transcriptional RNA processing, protein modification, and DNA replications. Okay, for the disease association, so your Epstein-Barr virus try to cause here the following diseases. Number one, IM. Infectious mononucleosis is a lymphoproliferative disorder characterized here by the proliferation of your mononuclear cells. Again, your mononuclear cells could be your lymphocytes or macrophages, but in the case of your IM here, it's more of the what's proliferating here would be your lymphocytes. Okay, then we have your Burkitt lymphoma. So this is a malignant lymphoma of your lymphoid tissues. Then we have also here the carcinoma, nasopharyngeal carcinoma. We have also here the neoplasm of your thymus, parotid gland, and supraglottic larynx. Again, all of that one is associated with your Epstein-Barr virus. Okay, now we have here the signs symptoms of your Epstein-Barr virus infection. So again, this is being called here as your kissing disease because again, the virus here reside in your nasopharyngeal area and it try to affect almost 90% of the population. Very common among young adult populations. Okay, for the signs symptoms, most likely this one is asymptomatic, but others would have here the patient try to show signs symptoms, it become a flu-like symptoms. The flu-like symptoms includes the fever, headache, malaise, sore throat. We have also here the cervical lymphadenopathy, splenomegaly, and we have your hepatomegaly. Okay, we have here the laboratory test for your Epstein-Barr virus infection. So hematology, we have here leukocytosis. So that's 10 to 20,000 increased WBC count. Differential count in the early infection characterized by the increase in your neutrophilia. But later on, as the disease progress, it become here lymphocytosis, which 60 to 90% of the entire differential count is made up of your lymphocytes, of which 5 to 30% of that is reactive lymphocytes. Another test, this one is heterophile antibody positive. Again, this would differentiate this one from your cytomegalovirus because in the cytomegalovirus, it's heterophile antibody negative. Then we have also hepatitis manifestation with an increase in the liver enzymes. Okay, then we have here the immunologic manifestation related to your Epstein-Barr virus infection. So, first we have here the heterophile antibody. We define heterophile antibody as the broad class of antibody that cross-react with the different surface antigen from the different mammalian species. Heterophile antibody could also be present even in the normal population, only that the titer is less than 1 is to 56. Okay, but if your titer here is more than 1 is to 56, then it would diagnose here that you are positive for the Epstein-Barr virus. Heterophile antibody could be produced here not only by your Epstein-Barr virus, could also be produced here in the Forceman antigen or even your serum sickness. Okay, to test here the presence of your heterophile antibody, we have here your Paul Banel test. So, since you're detecting for heterophile antibodies, which are your antibodies, dapat ang reagent natin ay antigen in the form of your sheep red blood cells. So, again, in the presence of these three, because all of them are producing heterophile antibody, so all of them will give a positive result in the Paul-Banel test. Again, ang magpapositive sa Paul-Banel test are those having heterophile antibodies. Okay, to differentiate, since all of the three here are producing heterophile antibody, we could not differentiate them as kung ano ba talagang sakit ni patient. So, differentiate that one, we have here your Davidson differential adsorption test. Okay, based on the principle here that your Epstein-Barr virus is adsorbed only by your ox beef, but not your guinea pig kidney cells. Forest-man antigen is adsorbed by the guinea pig kidney cells, but not your ox beef. And serum sickness is adsorbed by both your guinea pig kidney cells and your ox beef. Ang procedure nito, ganun lang, prepare ka ng dalawang test tubes, tapos ito ang kuha ka ng sample ni patient, i-add mo itong dalawa. Okay, then after that one, you try to centrifuge. So, pag sinabing adsorb, okay, for example, kung ito ang ating tube here, so after centrifugation, pag na-adsorb siya, dito siya magiging part na siya ng precipitate natin. Okay, you take the supernatant. Kung na-adsorb siya, like for example this one, pag na-adsorb siya dito, ang supernatant natin, expect mo na nawala na siya doon. It's because since na-adsorb siya, dito na makikita ang ating antibody. So, ang supernatant mo, wala na siya. Just in the case, for example, pag hindi siya na-adsorb, meaning to say, still andun pa rin siya sa supernatant natin. To check for that, okay, then you need to perform another test with the use of your indicator cell. Okay, take the supernatant, then try to check with your indicator cells in the form sheep red blood cells. Notice here in your Epstein-Barr virus here, since dito na-adsorb siya, so pag na-adsorb, meaning to say, nawala na siya sa ating supernatant, and therefore expect here na pag nag-add ka ng sheep red blood cells, wala na siyang agglutination. In the case naman dito, since hindi siya na-adsorb, so andun pa rin siya supernatant, pag nag-check ka ng supernatant, still andun pa rin siya, therefore magkakaroon siya ng agglutination. Dito naman sa force-man antigen, dito na-adsorb siya. So, pag na-adsorb, nawala na siya sa supernatant, therefore magkakaroon na siya ng negative agglutination reaction. Here naman, hindi siya na-adsorb, so doon pa rin siya, magpapositive siya. Dito sa serum sickness, dalawa sila na-adsorb, and therefore, if you to check the supernatant, wala na siya doon sa supernatant, and therefore, if you add your sheep red blood cells, wala na siyang agglutination. Okay, then we have also here the rapid test for detection of your heterophile antibody. So we have your monospot test. Okay, the monospot test will have the same principle as your Davidson, only that ang pinagkaiba ang indicator cells na ginagamit. Indicator cells here would be horse red blood cells instead of the sheep red blood cells being utilized in your Davidson differential absorption test. Same test result pa din naman sila. Okay, now we have here the serological manifestation. Okay, so again, 10 to 20% of the population which are positive for Epstein-Barr virus here do not produce a heterophile antibody. So for that, you need to test the patient here by serological panel. Aside from that one, you could also test here for those who are immunocompromised. Okay, we have here the serological markers. So we have here different antigen antibody. The first one, we have here the VCA, the viral capsid antigen. It could be found in the cytoplasm. So corresponding antibody, we have your anti-VCA IgM antibody. So this is antibody that is being produced the earliest time. But the problem with that antibody is try to disappear immediately. Okay, on the other hand, anti-VCA IgG antibodies being produced here 4 to 7 days after the infections. And once you produce antibody, or once you are infected, you produce this antibody, and this antibody persists for the lifetime. Andun pa rin siya, signifying that you have a past infection with the Epstein-Barr virus. Another marker, we have here your early antigen divided here into your diffuse and restricted. Diffuse could be found here diffusing in the cytoplasm and in the nucleus. And the restricted, on the other hand, could be found only here in the cytoplasm. This is not a reliable antibody marker here is not a reliable marker for diagnosing an acute infection. Anti-early antigen diffuse IgG antibody could be positive for acute infection, but the early antigen restricted IgG for adult population, even they have an acute infection, they are negative for that. But for the children, again, they will give a positive result here in an acute infection. Therefore, this one is not a reliable indicator for diagnosing an acute infection. Okay, the next marker we have here, the Epstein-Barr nuclear antigen. So this is being produced here, could be found in the nucleus of your B lymphocyte infected cell. So again, this is being produced here after the incubation period. As soon as the T lymphocyte try to destroy the B lymphocytes which has been infected by the virus. Antibody to this one, the anti-EBNA IgG antibody, signifies a past infection. At the same time, it's also being increased here or being produced as well in the nasopharyngeal carcinoma and even in the Burkitt lymphoma. Okay, and lastly, we have here the clinical correlations of the different serological markers. Okay, so if the patient's negative for all the markers here, so means the patient is susceptible. Pag sinabing susceptible, the patient would have the tendency to be exposed. On the other hand, in the acute infection, the patient would have positive for the VCA-IgM, VCA-IgG. We have also here early antigen diffuse IgG. And we have also here your heterophile antibody positive on patient, signifying that the patient has a recent infection. If the patient has been infected in the past and the patient has been recovered already, itong marker na lang makikita natin. Again, anti-VCA IgG antibody because again, this is a marker try to persist for the lifetime. And we have your EBNA IgG antibody. Notice here that their heterophile antibody is negative for that. That would signify the patient is currently do not have the infection. But if all the markers are positive, it would signify the patients to have an infection in the past, so nagkaroon siya ng reactivation, or it also signifies a reinfection. Okay, so that's all for this topic.